After DNA double helix, Crick worked with many scientists, including Cindy Brenner on central dogma. And eventually, their work led to the genetic code. I worked as a chemist in U.S. National Lab, Pacific Northwest National Lab, for 7 years on single molecule spectroscopy and microscopy, which that Harvard offered me a job in 1998. So upon arrival Harvard, I had a hunch that the single molecule approach would be very powerful for biology, but I had to learn biology, molecular biology, as a tenured professor. So I audited Rich Losig's molecular biology course. He was a wonderful teacher. I went with my students. Listening to his lecture on the lac operon, classic system that Tom mentioned this morning due to Jacob Monod and Huber, who did the work when he was a youngster at Harvard, sequenced the footprint of lac repressor, and his competitor, Mark Potashny, also a youngster. Actually, Tom worked with him for a few years, and Mark wrote these 2 books, which I read page to page at that time. Transcription factors control gene expression. So I sort of gave ourselves a homework to monitor gene expression in a live cell on a single molecule basis. We labeled the LAC-Y, the lac repressor controlled, see this in this operon. Each transcription factor controls several genes, the LAC-Y, LAC-A, right? So we put the GFP on the LAC-Y that goes to the membrane. So once the GFP gets on the membrane, the fluorescence is localized. We could see the single molecules. So this was the experiment of JHL and EG. In this overnight movie, we observed protein production, 1 molecule at a time. And Long Kai and Neil Freeman, Long is in the audience, had a different system. They work out the theory. Don't have time to go into the theory. Basically, we were able to quantitatively describe the central dogma of molecular biology in bacteria. Johan Elf, working with Jimmy Lee, labeled lac repressor, so if it binds there, then you see this bright spot. Again, we did this with the approach detection by localization. If it's localized on the DNA lac repressor, then you see it, just like the membrane. And if there's sugar in the media, then it's gone, right? So we were able to see all of this quantitatively in a live cell. And then, that was the repressed condition. Under induced condition, it's still stochastic. So I think gene expression is really a single molecule problem because there is only 1 copy or 2 copies of a particular gene. Hence, it is intrinsically stochastic. So that's the bacteria case. So this was actually my only paper with Tom. Tom was a colleague over MCB. We started to work on mammalian cell, and I learned it's totally different. In a bacterial cell, the transcription factor acts like a key to turn on and off a particular gene by repression. That binding time of lac repressor, 40 minutes. When we start to measure in mammalian cell, just a few seconds. And then we quickly realize, people have been saying this, there are multiple transcription factors, it's a combination. It's like a key chain. To solve this problem, we need new technology. So my group, in the past 10 years or so, has been working on single-cell genomics. It is fair to say that due to the contributions of many groups, single-cell genomics has changed the way biology and medicine are being conducted. Fu-Chou Tan's work with Kai-Chin, and he mentioned in 2009, first single-cell transcriptome using next-generation sequencing. My group did Malbec single-cell genome amplification in 2012. Fu-Chou, after he came to Biopic, did the first single-cell methylome, and Will Greenleaf at Stanford did the first chromatin accessibility, and very quickly became single-cell technique as well. And Peter Fraser in UK at that time did the first single-cell 3D genome. And in 2018, we did the DeepSeq that allow us to obtain the first 3D genome structure of a single human cell. You see these 46 chromosomes, 23 from each parent. Each one is folded. You have to do single cell because otherwise you just get the average of a particular chromosome. But one is surrounded by 6, say, another cell will be another 6. Okay, so I just want to mention this is not done by imaging. Okay, so it's done by sequencing. And compared to the super resolution microscopy method, the spatial resolution is comparable, but we get the whole field of view of the entire nucleus. We use the chemistry due to Joab Decker, who was at Harvard. This first paper, 2002. Recently, we use the micro-C instead of restricting enzyme. So this is chemistry developed by Oliver Rundle at University of Massachusetts. It cuts the DNA in between nucleosomes, so the spatial resolution is high. But I want to mention that for single cell genomics, we many groups here benefit from this paper. Biochemists isolated a study, TM5 transposase. Will Greenleaf and I independently took notice of this. Actually, Steve Quick used this to prepare the library. Most of you know Will did a TAC-seq. His sequencing cost is 1% of mine because we were working on the single-cell genome. So Lienti is a whole linear amplification method, but it did pay off for us for the later on, the single-cell 3D structure. Yan Yi and, actually he didn't talk about this. The single-cell transcription also with the transposase, with Jianbin. And Richard, I'm not sure if he'll talk about this. This was actually, he was trying to do this as a graduate student at Harvard, but the paper finally about to come out because he was busy with COVID-19. Once you have the 3D structure, so you can also have the simultaneous methylome, right? So the green is the euchromatin, and the purple is the heterochromatin, which is unmethylated in the gene body, okay? There's a lot of discussion about phase separation. So here we see the clear phase separation of the euchromatin and heterochromatin, but a lot of these microscopy papers about phase separation, they say, oh, I saw like oil droplets in water. But you see, when we section through this, there's only 2 phases. All this red and purple, they're connected, just one phase. I mean, this is not news. And people doing X-ray microscopy knew this a long time ago, right? So as far as we can see, 2 phases, euchromatin and heterochromatin. Now, some histones are modified, which would have different affinity for certain transcription factors. But to imagine that you have all these different droplets, how complicated it would have, I think God wouldn't do that. It's just too complicated. So in this euchromatin space, every transcription factor has one concentration. Fundamental difference between the mammals, the bacteria. In the bacteria, one transcription factor controls several genes in the opera. In human genome, each protein, one gene, okay? But in the promoter region, their transcription factor is a combination. So really the question is how the gene regulation is done, okay? Because this is a single molecule problem. Each gene is fluctuating. If you ask this question, if I look at a pair of genes, do they go like this, correlated or not correlated? See, you should be able to do this with a single-cell data, right? Except you need a high detectability. We improve the detectability. We have a so-called Malbec DT, and we see in this covariance matrix, this is about several thousand genes, expressed genes, and this is several thousand expressed genes. When they are correlated in time, it's red color, okay? So we zoom in, what do you see? You see these squares. Okay. So these genes, say 4,000 genes, they're like a troop, okay? And the soldiers in different units, they need to get up together, go to bed together because they must fight together, right? So this is the protein synthesis module. You look at the genes, I mean, it's obvious. They're all involved in protein synthesis. So each one is fluctuating, and we know this, the case, many microscopy experiments. So this is actually a transcription factor. This is Mike Weiss data, almost 20 years ago. You look at the cycle, half an hour, 1 hour, right? So because gene expression is a single molecule problem, it's never constant. It goes up and down. But if this protein-protein interaction, in order to finish a biological function, you need proteins to work together, tens of them. In this one, we got maybe 60 of them work together. Okay, they all have their own promoter region, right? So really the hypothesis is the combination of transcription factors that regulates the protein modules. We call this protein modules, and this has been confirmed by single cell mass spec as well. We just had a paper, okay. So now the next question is, where are these genes? Are they at different chromosomes? Yes. They're not on the same chromosome. Okay, now since we have the 3D genome, right? So spatially, are they correlated? So this is the protein synthesis module, and these genes, if it's white color, that means they're very close. You see a few of them are very close. That's because they're on the same chromosome. So on different chromosomes, they really don't, there is no such, we don't have strong evidence for the so-called factories. I mean, there are enhancers, don't get me wrong, I'm not against enhancers, but all these genes, if they have protein-protein interaction, they come to the same factory, we don't see that, okay? And we don't have strong evidence whatsoever for this phase separation for gene expression. Many researchers talk about in recent years. Now this is popular T-SNE plot. Each dot is a cell, right? Here, each dot is a gene. And you really see the gene clusters, and these are the modules, and this red one is the protein synthesis module. And we want to know the transcription factor that control each protein. Now, in the audience, there's one man who pioneered the technique to see the transcription factor bindings through antibody, and you pull down, you do sequencing. That's Renbin Cheap. ChIP-seq contribute a lot to our understanding, particularly the encode project. There's also DeamInasE, not as sensitive with the technique that I'll be telling you about. All right, we found double-strand DNA DeamInasE. So we found that from nature. It's a toxin molecule, right? We got structures. So basically, it converts C to U, and U reads like T. So we can do footprinting. We'll be looking for this in the past 5 years or so. Finally find one called DDDB. So in bare DNA, it's a very good yield and single cell. This is cellular data. This high yield is important for single cell. Okay, so our technique is called footprinting by DeamInasE, FOODIE. We run out of names for red vines. So basically, the open region that you see with ATTAC or Greenleaf's approach, in the open region, if transcription factor binds to DNA, you will leave a footprint. Then the C will not be converted to U, reads like T. So we would have single base resolution, and the DNAs could only do cell lines. We want to be able to do single cell. Basically do cell typing and look at the transcription factor of a pure cell type, right? We want to do it in situ, and we want high throughput. Let me just quickly show you along the genome. So we have a visualizations. So this is the along the genome, and you see this is the attack signal, and you see these two nucleosome, these steps. Basically, you've got nucleosome, you have no conversion, and this is +1, +2, -1, -2, and then we see the footprints of 3 here. I want to emphasize, even though this is, each cell only have 2 alleles, right? So we do experiment on a few hundred, a few thousand cells, but the raw data is a single molecule data because it's a DNA that is, say, 250 base long, right? So on there, we can see the red is Watson, the green is Crick. 2 complementary strands to see if they're converted, we label it red or green. So basically, we have the thermodynamic binding fraction, okay? So we can go anywhere. So I'm driving along the chromosome 1. So in the middle, not too much stuff. Let's go quickly. And then when you get close to a gene A, then you start to have the open region, lots of data. And then, basically, that's the data. And we'll be analyzing it in the past few months. We can look at not only the equilibrium binding fraction, but also the cooperativity, because we can simultaneously, you see 2 prints, C converted, 2 positive and positive, negative, and 2 negative. So this allows us to calculate this phi coefficient. So back in 2013, we had a paper for 6 biopic professors. Tom probably remembers this. He was our SAB chair. So basically, we found allosteroids through DNA when these 2 DNA binding proteins are close to each other, but they're not touching, okay? They can positive cooperativity or negative cooperativity. Not like one go up, one go down. Either go up together or go down together. This is dictated by thermodynamics. Okay, we found that if we just look at the pairs, there are a lot of positive cooperativity, and there's a large effect, a factor of 10 and higher. And this is because nature selects the sequence. Okay, we found very few negative cooperativity, and we found in the promoter region, the average transcription factor, so 5 proteins. And the tissue-specific, the average is slightly lower than the housekeeping. I mean, we're still in the process of decoding this to figure out the regulation, the principle or the rules behind this. I mean, obviously it's not random, but we didn't have the information to look at this. If you believe the hypothesis, it's the combination. I mean, why is that? It's because there are 20,000 genes, 80,000 genes get expressed, say, but there are only, it's usually less than 800 transcription factors. So it's definitely not one-to-one, as in the case of bacteria, right? So, but if you have combination, you could have more. But so no genes would have the same, right? So we need to know this combination in order to decode this. We now have a different cell line for these different tissues. There are shared ones, and there are also this cell type specific. For doing the summary, I think you get the idea. We got a technique. We also have an assumption, and I hope the analysis of this new kind of data will teach us a lot about how in mammalian cells, gene expression and protein-protein interaction are regulated. And I want to thank my group members working on this. I gave a structure point of view, but I must say electron microscopy and X-ray crystallography has contributed a lot to the understanding of transcription and translational machineries involved in the central dogma, and that will be the topic of the next talk. We have excellent young scientists. Thank you.